meiosis II. What happens in meiosis I is that diploid cells become haploid cells. This is a process referred to as reductive division. Diploid cells have two copies of each chromosome, and therefore they are often referred to as 2N. Haploid cells have one copy of each chromosome, and therefore they are often referred to as 1N. In meiosis I, homologous chromosomes separate, so let's talk about what that means exactly. At the bottom left side of the screen, I've drawn a diploid cell. This cell has two copies of every chromosome. However, for simplicity's sake, I've only shown one particular chromosome. Let's imagine that this is chromosome 21. Every one of our chromosomes will go through the process on the screen, but we're going to focus in on it in detail for one particular chromosome. For argument's sake, let's say it's chromosome 21. So this is a diploid cell which has a paired chromosome 21, or in other words, has two homologous copies of chromosome 21. One of those copies, denoted by the blue line here, came originally from the father. That's the paternal copy of chromosome 21. The other copy, denoted by the red line, came originally from the mother. During interphase, the two chromosomes will be replicated and they will become two chromosomes with sister chromatids. This is the paternal copy of chromosome 21. It has two sister chromatids attached together. The same is true for the maternal copy. The process of meiosis I then begins, and the first thing that occurs is there is some crossover or recombination of genetic material. So the two chromosomes, the one originally from the father and the one originally from the mother, exchange some genetic material. They then go through a cell division and become haploid cells that are only 1N. These cells that result from meiosis I have a single copy of chromosome 21, and that copy is a mixture of the chromosomes that originally came from the father and the mother. In the second step of meiosis II, the sister chromatids separate and form four daughter cells. So here we've got two cells which have sister chromatids, and they separate and create four gametes, and all of these cells are 1N, meaning they have only one copy of each chromosome. And here's a summary slide of the entire process. You begin with a cell with two copies of each chromosome. During meiosis I, the homologous chromosomes separate and undergo crossover and recombination. During meiosis II, the sister chromatids separate to create four gamete cells that are 1N. So that is how the process of meiosis plays out in general. Now let's talk about exactly how it occurs in the sex organs of males and females. In males, the process of spermatogenesis generates haploid gamete cells. This process begins at puberty. Males have cells called spermatogonium, which are diploid or 2N. They undergo mitosis, not meiosis, to become cells called primary spermatocytes. These cells are diploid and have both an X and Y chromosomes, just like all the cells in the male. The primary spermatocytes undergo meiosis I, and they will become cells called secondary spermatocytes, which are now haploid and have only an X or a Y chromosome. Those cells will then go through meiosis II, and they will become four spermatids, which are the cells which contain the separated sister chromatids. They are haploid cells. They contain only an X or a Y chromosome, and then those cells go through spermiogenesis and they become spermatozoa or sperm, which can then go on to fertilize the egg. In females, the process is different. The generation of gametes is called oogenesis. It begins with primary oocytes, which are formed in utero in the female. These are diploid cells, and they are just beginning the process of meiosis I, and they are arrested in prophase of meiosis I until puberty. And this is a very important point. Meiosis is very drawn out in females, and for that reason, as we will see later, this process is vulnerable to errors that can lead to pathology in the offspring. At puberty, a few of the primary oocytes will complete meiosis I each menstrual cycle. Some will form what's known as polar bodies and degenerate. Others will form secondary oocytes, which are haploid and can go on to become eggs. At this point, meiosis II begins but arrests in metaphase. If fertilization occurs, then meiosis II will become complete. So once again, the key point here is that this is a very drawn-out process in females, and therefore it is more vulnerable to errors than the process in men. Now that we've discussed meiosis, for the rest of this video I'm going to talk about errors in meiosis that lead to pathology, and let's begin by defining an important term, and that is the term aneuploidy. Aneuploidy means an abnormal chromosome number, either an extra or missing chromosome within a cell. Disomy means two copies of a chromosome. This is the normal state. Monosomy means a cell contains only one copy of a chromosome. This is a form of aneuploidy. Trisomy is also a form of aneuploidy. This means a cell contains three copies of a particular chromosome. Meiotic non-disjunction is a failure of chromosome pairs to separate during meiosis, and it is the most common mechanism of aneuploidy, and it can occur in either meiosis I or meiosis II. If there is a meiosis I non-disjunction, the homologous chromosomes will fail to separate. This will generate one cell with no chromosomes, which will go on to generate gametes that have no chromosomes. The other cell will contain two copies of a particular chromosome, and it will go to form gametes that have two copies of a chromosome. Remember, these gametes are supposed to only have a single copy of each chromosome. They are supposed to be 1N. However, in this case, as you can see, they are 2N. They contain two copies of the chromosome that came from the parent. Importantly, note that when there is a meiosis I non-disjunction, the gametes have a mixture of genes. In other words, they have one copy of the chromosome that came from the father and one copy of the chromosome that came from the mother. This is an important feature of a meiosis I non-disjunction event, and it is different from what you see in a meiosis II non-disjunction. If there is a meiosis II non-disjunction, meiosis I will occur normally and the homologous chromosomes will separate. However, in meiosis II, the sister chromatids will fail to separate. This will generate one gamete which has no genes and another gamete which is diploid. But importantly, this diploid gamete has no mixture of the original paternal and maternal genes. Note that in the example I've drawn here, it's got two copies of the red chromosome which initially came from the mother. It does not have one copy of the red chromosome and one copy of the blue, and this is an important feature of meiosis II non-disjunction events. And here's a slide just to pull it all together. If you have a meiosis I non-disjunction, the two homologous chromosomes will fail to separate. This will generate gametes that are 2N and have a mixture of the paternal and maternal genes from the original individual. If you have a meiosis II non-disjunction, the sister chromatids fail to separate. This will create a 2N or diploid gamete that has two copies of the same chromosome from the individual in whom the non-disjunction occurs. And you'll note that I've not shown crossover and recombination here, and that's because crossover and recombination is rare in situations where non-disjunction occurs. Monosomy occurs when you have fertilization of a 1N or normal gamete with a 0N gamete that contains no copies of a particular chromosome. And we saw how in meiosis there were a couple of ways by which you could generate gametes that did not contain a copy of a particular chromosome. This can lead to monosomy for that particular chromosome. Most monosomies are not viable and will result in a spontaneous abortion. Turner syndrome is one example of a monosomy. It's a monosomy of the sex chromosomes where a girl is born with only one X chromosome. Trisomy occurs when you have fertilization of a normal 1N gamete with a 2N gamete, and we saw a couple of ways by which meiotic non-disjunctions could lead to gametes that contain two copies of a particular chromosome. Most times when there is trisomy, it's not compatible with life and will lead to a spontaneous abortion. However, there are a couple of exceptions. Trisomy 21 is viable and it will lead to Down syndrome, and 95% of cases of Down syndrome are due to meiotic non-disjunction. Trisomy 18 will also lead to Edwards syndrome, which can be viable, and trisomy 13 can lead to Patau syndrome, which also can be viable. Maternal meiosis I non-disjunction errors are a common cause of trisomy. As we discussed earlier in the process of oogenesis, meiosis I is protracted in females, begins prenatally, and it's completed at ovulation many years later. This leaves lots of time during which non-disjunction can occur. In addition, this is the reason why advanced maternal age is a risk factor for many forms of trisomy. With advanced maternal age, the eggs that are being released have spent longer going through meiosis, and therefore there's more time for non-disjunction. When a baby is born with trisomy, the cause of the non-disjunction that led to the trisomy is suggested by the trisomy genotype in the child. To understand this, let's imagine that we have a father with two copies of chromosome 21, and we use genetic techniques to identify unique features of each chromosome 21, and we call one of the father's chromosomes 21A, and the other one we characterize and call 21B. We do the same thing for the mother and characterize her two chromosome 21s as 21C and 21D. Now if we look at the baby with trisomy, let's suppose the baby has trisomy 21 and has copies of chromosome 21 that are A, C, and D. Well, we know this must have been an error in the mother because there are two copies of chromosome 21 that came from the mother. In addition, we know it must be a meiosis I non-disjunction event because the homologous chromosomes in the mother, C and D, failed to separate, and that's what happens when there's a meiosis I non-disjunction, as we discussed earlier. Conversely, let's suppose that the baby had trisomy 21 and had chromosome 21's A, C, and C. Once again, we would know it is a maternal error because there are two copies of chromosome 21 from the mother. In addition, we would know it's a meiosis II non-disjunction because in meiosis II non-disjunction, the sister chromatids fail to separate and you get two copies of the same chromosome in the fetus. Uniparental disomy is when a child inherits two copies of one parent's chromosome and no copies of the other parent's chromosome. This is a rare event. There have to be abnormal gametes from the male and female. One of them has to contain no copies of a particular chromosome and the other one has to contain 2 copies. Once again, you can use the genotype of the fetus to figure out where the non-disjunction event occurs. For example, once again, if we have a father with 2 chromosome 21s A and B and a mother with 2 chromosome 21s C and D, suppose that the child is born with AA. We know that the child must have uniparental disomy coming from the father because the child has 2 copies of the father's chromosomes. In addition, we know it must be a meiosis II error because the child inherited 2 copies of the same chromosome from the father, and this is what happens when there's a meiosis II error. This is called isodisomy. It's uniparental disomy with 2 copies of one chromosome. Conversely, suppose the child was CD for chromosome 21. This would be a meiosis I error in the mother. This would indicate that the homologous chromosomes in the mother failed to separate, and this is called heterodisomy. A child born with uniparental disomy is euploid, meaning it has a normal number of chromosomes. There is no aneuploidy, and usually it has a normal phenotype. The one problem that can result from uniparental disomy is that it can lead to a phenotype for a recessive disease. For example, suppose the father was capital A, and little a is a version of a gene that is recessive for a disease. If uniparental disomy occurs and the child inherits 2 copies of the diseased gene, that child will get the disease from a single parent, not in the usual way that recessive disease is inherited, which is from 2 parents who are carriers. The last topic I'll discuss in this module is that of a Robertsonian translocation. This occurs when there is fusion of the long arms of 2 chromosomes. This happens to chromosomes that are known as acrocentric. These chromosomes have a centromere very close to their end, and thus they have a very small short arm. This is true for chromosomes 13, 14, 21, and 22. To understand a Robertsonian translocation, let's look at this picture I've made on the screen. So we have a normal chromosome 14 and a normal chromosome 21. If these 2 chromosomes undergo a Robertsonian translocation, the long arms will link up and create a fused chromosome 14 and 21. The short arms of both chromosomes will be lost. This usually carries no important consequence for the individual in whom the translocation occurs. It's a small amount of genetic material that is lost, and it usually does not lead to any clinical consequences. The major implication of this translocation is that the person carrying these 2 fused long arms is likely to have many offspring with chromosomal anomalies. To understand this, let's imagine that we have a normal father who has 2 copies of chromosome 14 and 2 copies of chromosome 21. And let's imagine that he mates with a mother who has undergone a Robertsonian translocation. So she has 1 normal copy of chromosome 14, 1 normal copy of chromosome 21, and 1 fused chromosome that resulted from the Robertsonian translocation. At the bottom of the screen, I've shown all the potential zygotes that can result from this union. The father is always going to donate 1 copy of 14 and 21 because he is normal, but there are many different possibilities for which 2 chromosomes the mother will donate. If the mother's very lucky, she will donate her 2 normal chromosomes 14 and 21 and the child will be normal. If she's a little bit less lucky, she will donate her fused chromosomes, and this will create a child who is a carrier for the Robertsonian translocation. All of the other possibilities are very bad for the fetus. If the mother donates her translocated chromosome and her normal copy of 21, then the baby will have trisomy 21 and will have Down syndrome. And a Robertsonian translocation is one mechanism by which Down syndrome can occur. It's not the most common, but it's one possible mechanism. All of these other possibilities, including monosomy 14, monosomy 21, and trisomy 14, are not going to be viable, and therefore this couple is likely to have many failed pregnancies and many spontaneous abortions. So on this slide, I've summarized the important points. The carrier of a Robertsonian translocation has only 45 chromosomes. 1 is a translocated chromosome. The carrier has lost the short arms, but they will usually have a normal phenotype because there's no clinical consequence from loss of the short arms and therefore no disease. 13 and 14 and 14 and 21 are the most common, and because 21 is often involved, this is one of the ways you can get Down syndrome or trisomy 21. The main clinical consequences are many monosomy and trisomy gametes, and for this reason there are frequent spontaneous abortions. The other consequence is that the carrier may have a child with Down syndrome or trisomy 21. Because of the possibility of a Robertsonian translocation leading to fetal loss, a karyotype is a test that's often done in couples who have recurrent fetal losses. A karyotype is used to diagnose chromosomal imbalances. A karyotype creates a karyogram like is shown on the screen here, and you can identify 2 fused chromosomes from a karyotype. So this is a test that's often sent in the workup of infertility to see if one of the parents is a carrier of a Robertsonian translocation. And that concludes our video on meiosis.